Dr. Sage here. We're continuing our discussion of genetic analysis and genetic engineering. In today's video, we're going to talk about DNA analysis, maps, and profiles. By the end of this video, you should be able to outline in general terms the process of DNA sequencing, outline the general steps in DNA profiling, discuss the significance of single nucleotide polymorphisms, also known as SNPs, in DNA analysis, and describe the utility of DNA microarray technology. Analysis of DNA can reveal genetic abnormalities, ancestry, etc., determine if one sample of DNA is the same as another sample, for example, when a criminal is identified by DNA extracted from a hair left at a crime scene. Sequence maps are the most detailed maps of a genome. They give an exact order of bases in a plasmid, chromosome, or genome. Genomes of thousands of organisms have been sequenced, including viruses, bacteria, and eukaryotic organisms, including humans. Humans share about 80% of their DNA code with mice, about 60% with rice, and even 30% with the nematode, the worm C. elegans. Here's a brief overview of how DNA sequencing is conducted. First, a dideoxynucleotide is similar in structure to a deoxynucleotide, which is typically integrated into the DNA, but is missing the 3' hydroxyl group. When a dideoxynucleotide is incorporated into a DNA strand, DNA synthesis stops. Using dideoxynucleotides, which are tagged with fluorochromes, so they'll fluoresce a certain color, a mixture of DNA fragments of every possible size, varying in length from one nucleotide, can be generated. The DNA is separated on the basis of size, and each band can be detected with a fluorescence detector. The following are added to a PCR reaction tube: DNA template, primers, DNA polymerase, deoxynucleotides, and fluorescently labeled dideoxynucleotides. At each location, either a deoxynucleotide is added and the elongation continues, or a dideoxynucleotide is added and the elongation stops. This process results in fragments of all sizes, each with a different fluorescently labeled end molecule. The fragments are then run through a capillary gel, where light from a laser passes through and the detector detects which fluorescent dye is passing through. A computer then identifies each band as it passes by the laser. So doing this, we can determine the sequence of a strand of DNA. There's also the whole genome shotgun sequencing method. This begins when the whole genome of an organism is broken down into smaller, manageable fragments. The fragments are separated through gel electrophoresis, and each fragment is inserted into a plasmid and cloned into an E. coli cell. Plasmids are purified, and DNA fragments are sequenced by automated sequencers. A computer program then takes all the sequence data and is able to find where the sequences overlap. Contigs are larger continuous set of nucleotide sequences produced by this automated process. Contigs are put in the proper order to determine the entire sequence. Then a human manually examines the sequence, looking for irregularities, frame shifts, and ambiguities. With genomes being billions of base pairs long, we want to also involve bioinformatics. This allows us to manage DNA produced through high throughput screening. It allows us to analyze and classify genes, determine protein sequences, and ultimately determine the function of genes. Annotating the genome involves determining the functional information of a gene sequence. Well-annotated genomes will provide a complete understanding of normal cell function, disease, development, and aging. Well-annotated genomes will allow us to characterize genetic mechanisms behind pathogens and allow development of new treatments. DNA profiling, also called DNA fingerprinting, is a tool of forensic science developed in the 1980s, based on the principle that the way nucleotides are combined is different for each organism. It is used to differentiate between organisms that might be causing a disease outbreak, and is used in criminal investigations to match a suspect to the DNA left at a crime scene. In DNA profiling, primers bind to repetitive sequences found at multiple locations in the genome. These regions are amplified using PCR. Gel electrophoresis is used to identify the sizes of the amplified regions, and the amplified fragment patterns can be compared to known samples to identify the strain. RFLPs, or restriction fragment length polymorphisms. Restriction enzymes cut DNA-specific restriction site. If a given strand of DNA possesses the restriction site, it will be cut into two pieces. If the same strand from a different person does not have the restriction site, it will not be cut and remain a large piece of DNA. So for example, let's say we have an individual that has these three restriction sites for a specific restriction enzyme. In that case, it will cut this piece of DNA into the four pieces. However, if another individual has a mutation that changes the restriction enzyme site located here, so the restriction enzyme will no longer cut here, then instead of having fragments of these sizes and four fragments, you would then have three fragments of different sizes right out of the gel, and you can identify the differences between them. Single nucleotide polymorphisms, also called SNPs, are important individual traits. Only a single nucleotide is altered. Tens of thousands of these differences at a single locus exist throughout the genome. Scientists are mapping SNPs to determine which ones put humans at greater risk of disease. Many companies will map SNPs and give a person an estimated risk for a variety of diseases. DNA microarray analysis allows biologists to view the expression of genes in any given cell. This tracks the expression of thousands of genes at once. So, for example, let's say you want to know what are the differences in gene expression between healthy cells and cancerous cells. What we do is we take these two cell types and extract their mRNA from them. We then take the mRNA and use reverse transcriptase to build cDNA from the mRNA, and then we label these cDNAs with a fluorescent probe. So for example, the ones from the healthy cells will be tagged with a green fluorescent probe, they'll glow green, and the cDNA from the cancerous cells will be tagged with a red fluorescent probe, so they'll glow red. Then you have a microarray, which is a little silicon chip looking kind of like a computer chip. What it has is thousands of spots of DNA on it. Well, when you apply the cDNA to the microarray, the cDNA, which came from mRNA, which remember are genes that are being expressed, will bind to the particular places on their microarray that correspond to certain genes. And then you can compare how the gene expression is different between the healthy cells and the cancerous cells. For example, this is an image taken from a microarray, and all the genes that are highlighted in green are only expressed in healthy cells, whereas the ones highlighted in red are only expressed in cancerous cells, and the ones that you can find in yellow are expressed both in cancerous cells and in healthy cells. So by performing this microarray, we can determine which genes are expressed differently in cancer versus in healthy cells. Microarrays can be used to develop sensitive diagnostic tests that search for a specific pattern of gene expression. For example, being able to identify a subtype of cancer, allowing doctors and pharmacologists to treat each cancer with a drug that will be most effective. Alright, this was your introduction to DNA analysis, maps, and profiles. Until next time, this has been Dr. Sage.